Dear Hans, dear Alberico, thank you very much for the kind invitation to speak to you today on Genome Wide Association Studies and what they tell us for the pathophysiology of coronary artery disease. I have no disclosure. This is the traditional view on genetics, where we learned a lot from monogenic disorders and how mutations affected proteins and the pathophysiology that is associated with these proteins. But nowadays we focus much more on complex genetics because we learned recently that complex genetics have, on a population level, a far larger implication than monogenic disorders. And here there are not only variants sitting in the protein coding region of the genes, but also in regulatory regions, and there may be also interaction with exogenous factors that affect the complex pathophysiology that is driven by genetic as well as environmental factors. We appreciated the implications of these many complex variants by the era of the Human Genome Project, where many variants were identified that contribute to the way we look like, but also contribute to the predisposition that we have for diseases like atherosclerosis. And the way to study this is by Genome Wide Association Studies, where thousands of patients are compared with thousands of controls from chromosome 1 to chromosome 22. There are millions of these genetic variants that can be tested in an association approach, where we look whether one of these or many of these have different allele frequencies in the patients and in the controls. Here you see again the chromosome 1 to chromosome 22, and each of these dots is referring to one of these single nucleotide polymorphisms, exchanges of nucleotides, and their association with disease. Here you see the p-value by which these individual variants associate with the risk to disease. Because there are so many variants, we have to be very stringent in terms of the significance level, and nowadays we consider p-value to be significant at a level of 5 times 10 to the minus 8. And we can only be sure if this is a real finding if we can replicate this in other populations. So this is the overall design, and just to illustrate how important numbers are to come to significant results, here is the most prominent locus that has been identified by this technique, the locus on chromosome 9p21. And this is here the significance level in 1,000 patients and 1,000 controls. If we increase the numbers of cases and controls, you can see how a peak discriminates with fairly high significant levels, astronomical actually significance levels, but all this is only brought about by studying large numbers of cases and controls. So this has been done over the last 10 years, and we are kind of proud that we conducted the first genome-wide meta-analysis on coronary artery disease, and that came up with this particular locus at chromosome 9p21, but also some other loci. So this was the situation about 10 years ago when 3 genes or chromosomal loci met the level of significance, and this is what evolved since then. By now we are about to 60 chromosomal loci that all are associated with coronary artery disease at this particular magic level. And just to make sure, ABCG5 and ABCG8 are amongst these loci that met the level of genome-wide significance for coronary artery disease, telling us the importance of this particular gene when it comes to the risk of coronary artery disease. Actually, I mentioned how important numbers are in finding these genes. Here you see again numbers of individuals that have been studied by this particular technique, and the number of chromosomal loci that met this particular level of significance. You see a clear-cut linear correlation, and that implies that there may be actually many more chromosomal variants that affect the risk of coronary artery disease when we have the opportunity to study more and more individuals. Now, I guess the first conclusion that we can safely draw from these data is that hundreds of genes and chromosomal loci are involved in the pathophysiology of atherosclerosis. So what do we learn from this in addition? I guess we can find new insights on established treatment targets. We may identify new targets for treatment, and we may refine risk prediction. Now, for this, I guess we have to have a closer look on these loci. And one thing that we can see, fortunately for us, that the odds ratio that comes with the risk alleles is fairly small. But remember, there are many of these risk alleles, and the